Study on Rumex vesicarius extract as a green corrosion inhibitor. This study was made by Messaouda Allaoui, Khair Rahim, Brahim Al-Abid, Ibtisam Ballawar. Abstract. In this study, the inhibition and adsorption properties of Rumex vesicarius flower, or so-called FRV, extract as corrosion inhibitors of XC70 carbon steel in 1 molary HCl were investigated using the weight loss method and other different electrochemical techniques like potentiodynamic polarization, electrochemical impedance spectroscopy. The corrosion rate was found to decrease with increasing inhibitor concentration, and the inhibition efficiency increases with increasing concentration of the extract, with the maximum percentage corrosion inhibition efficiency being 91.578% at 12.5 of volume. Polarization measurements revealed that the FRV acted as a mixed type inhibitor, and the inhibition efficiency of up to 92% can be obtained. Introduction. Several research and works have reported the use of plant extracts on the corrosion inhibition of metals in acidic media. These plant products are rich in organic compounds that can be extracted by simple processes. In addition, they are biodegradable, non-toxic, readily available, and renewable resources, as their use as corrosion inhibitors can be graded as an alternative to expensive and toxic synthetic compounds. Moving to the second part, which is the methods that was used in preparation of extracts FRV. The extract was prepared by soaking 200 grams of the plant powder in 5 liters distilled water solution for 24 hours. After filtration, add 1 molary HCl into 1 molary standard flask after adding little distilled water, and dilute the acid. Different concentrations were prepared as follows: 0.250%, 0.650%, 2.50%, 3.75%, 5%, 8.75%, 10%, and 12.50%, as illustrated in the following pictures. Moving to the electrochemical techniques, the first case, the immersion time was 30 minutes. The corrosion potential minus 480.2 millivolt in a frequency range from 100 kHz to 10 MHz by a parturition signal of 10 millivolt amplitude peak to peak at room temperature 23 to 24 degrees. For the second case, the immersion time was 40 minutes in open circuit at room temperature 23 to 24 degrees. The scan rate of 0.5 millivolt in the potential range of minus 750 to minus 200 millivolt. The results were obtained using the materials that will be mentioned here: computer-driven Voltalab 40 module, PGZ 301 potentiostat, galvanostat equipped with Voltamaster for software. Results and discussions. Using the weight loss method. Figure in 1. A. Variation in the corrosion rate, and B. in the inhibition efficiencies of carbon steel in 1 molary HCl at various concentrations of the FRV extract. For figure in 1.A also clearly reveals a general reduction in the corrosion rates in 1 molary HCl due to FRV extract addition. For figure in 1.B show that efficiency increased with FRV concentration. The protective action of biomass extract is often attributed to their phytochemical constituents with molecular and electronic structure. And now moving to discussing the results of the electrochemical methods. Figure 2 shows the inhibition efficiency increases with increasing inhibitor concentration, and the Nyquist plot semicircles with single capacitive loop and increasing diameter as the concentration of the plant extract increases. This indicates that the plant constituents are adsorbed on the metal surface, resulting in a decrease in double layer capacitance. For the second point, polarization studies indicated that the extract is mixed type, inhibiting both cathodic as well as anodic reactions, as shown in the illustration above. Moving to the last part with conclusion, the overall results led to the following conclusions. The corrosion rate decreased with increase in the extract concentration. The inhibition efficiency was found to increase with increased inhibitor concentration, with the maximum inhibition efficiency in 1 molar HCl was found up to 92% with 12.5% concentration of the inhibitor. For the second point, corrosion inhibition is mainly due to the adsorption of the plant constituents on the mild steel surface. Polarization studies indicated that the extract is mixed type, inhibiting both cathodic as well as anodic reactions.